developed for shellfish. But I'm going to make some comments about the benthic processes and models at the beginning here. But going through these models, they were developed for a benthic organism. But I think maybe I'd like to look at this in terms of a more general way in which models are evolving, and it goes to some of the comments we've heard this morning about how we go from these simple parameterizations, empirically based models in effect, to models that have more of a mechanistic basis to them. All right, so I am also going to talk a little bit about, in the context of these models, human impacts and management applications. And this isn't at the level of something like Atlantis or these end-to-end models. This is a specific application to look at a specific problem. And models are good for that. You don't need the full suite of everything in order to address an issue that may have relevance to society. Okay, so what I want to talk about here in the presentation, first talk a little bit about the benthic invertebrates, look at a population-based model, talk about the approach to that, modeling these benthic shellfish populations, and then the limitations of these approaches. Then a little bit about our individual-based model, the approach, combining it with the population model, and then genetic variability. All right? And Barish mentioned the fact that we're getting a lot of the genomic information. We're learning a lot about the genetics of organisms now. And if you really want to attribute cause and effect, you really have to get down at the genetics level. Also, it's through the genetic adaptation, the genetic evolution, that we're going to evolve or change in response to climate change, things like increasing pH, warming temperatures, and things like that. So we have to be able to deal with that somehow in the context of these models. And then just including remarks here. Okay, so benthic invertebrates, they're important parts of estuary and coastal food webs. They're important in general. A lot of them are chosen as sentinel species because they're long-lived. That's why we focus on shellfish in a lot of environments because of that. They provide coupling between the benthic and pelagic systems. Most of these, like the shellfish I'm going to talk about, have a post-settlement phase, which is sedentary, which is why they're a good sentinel species. And they also have a larval phase, which connects back to the pelagic system. All right. They filter water. They're important in ecosystem services way because of filtering water in bays and estuaries and coastal environments. And through filtering, they do things like regulate food concentrations. They actually can control the distribution of phytoplankton in areas. And they're also important in terms of nutrient cycling and returning nutrients to the water column. The other thing that's becoming increasingly people are aware of right now, things like shellfish are important to carbonate balances of estuaries. Right now, almost every estuary in the world has a negative carbonate balance, which means there's not enough shell in the estuaries, because when people harvest shellfish, you don't put the carbonate back in. All right, so I live in an area where the oyster shells and the clam shells are ground up and turned into sidewalks. All right, so if you're walking on shellfish sidewalks, that means your local estuary doesn't have those shells anymore. So this is becoming a problem with restoration efforts in trying to restore these estuaries to the previous forms. All right, and then the other thing is that many of these benthic invertebrates are commercially important species, and they're important to local history and culture. And I think Beth mentioned that earlier, or yesterday in her presentation, that a lot of this is cultural, what we value as a culture, and that's why we want to restore it, we want to save it, and maintain it. All right, so benthic invertebrates, if you look through the literature and you look at the models that have been done, usually for coastal and estuarine systems, and you look at the kind of model that Barish was just talking about, the NPZ types of models, there's usually a bottom boundary condition in areas where the bottom is shallow enough that it matters. All right, and the benthic invertebrates are parameterized in those models typically as a bottom boundary condition, usually in terms of nutrient supply back to the water column or through some filtration sink that just sucks things out of the water column. All right, so it's a loss of phytoplankton or nutrients. All right? And, or there may be a general benthic population that sort of grows and declines in response to various environmental conditions such as, bottom temperature, sedimentation rates, that kind of things. All right, the other type of model that's looked at sometimes is a coupled circulation biological model that usually focuses on the larval part of the animal's life history. So where do they go, where do they settle, and that's important in terms of maintaining populations, in terms of management. If you want to restore this reef, are you restoring the reef that is actually providing a source of larvae for the rest of the estuary, or is it the sink population, that kind of thing. And then there are some models that go all the way from circulation biology that includes the larvae and the post-settlement populations. And some of these are actually coupled to lower trophic level models. So there's a dynamically evolving pelagic system with a dynamically evolving food field, and you've got a real population on the bottom that's eating that and then contributing back through nutrients. So the message here is that there is there are a number of ways to do this, and a number of ways to put the benthic environment into these various kinds of models. All right, and all of them though, and what I hope to bring home as a message today, is if you're going to do this, you first have to understand the ecology, the physiology, and the life history of the organisms or organism that you're dealing with in terms of the benthic environment. And that goes to all of these. I think that was Barge's comment and various other comments we've heard from other people, is that I don't think anymore you can get away with just having the average phytoplankton or the average zooplankton. We're recognizing that species matter. And so that's why the emphasis now on actually understanding the life history and the biology of these species. Okay, a couple more general comments before I get into the slides here, into the models. Most models, the models I'm going to talk about, and even the majority of them, track energy usually in terms of calories, joules, or sometimes in terms of carbon. And for these species, that energy is given by the difference between assimilation and respiration. So what's left over is what goes into the animal. All right. Now, when you start dealing with things at the level of zooplankton and higher trophic levels, you have to deal with reproduction. It's particularly important like with the shellfish I'm going to talk about here. So you also have to not only grow the somatic tissue that the animal needs to maintain itself, you also have to produce the reproductive tissue along with it. All right. There's also mortality. It's natural mortality, predation mortality, and things like starvation that comes from lack of good quality food in many cases. The other one that was just mentioned is disease. All right, and this is a major problem right now in a lot of the estuarine and coastal systems, the shellfish and other types of invertebrates, a lot of, again, commercially important species are being impacted by disease. So there's a lot of work going on right now to understand that and understand disease transmission in marine systems, because the models we have for how we transmit diseases among ourselves don't work in marine systems. Those are density-based contact models. They don't really work for marine populations, and that's a whole other lecture I could give. But it's important, and I'll just put that out there as something to think about. Okay, there's also the pelagic phase of the life history. You need a circulation model when you start dealing with that. And then there are ranges of responses within species in their rates, like their respiration rate, assimilation rate. We don't all respire or assimilate at the same rate, and that's the result of genetic variability. Right? And that's something we want to build into these models now, because that variability is what gives the adaptation to changing environmental conditions. All right, and I'm going to, there are multiple kinds of species. Typically the models focus on the ones for which we have the data and the understanding, and that's what I'm going to do here is to talk about some oyster and clam models. Okay, now, if you have a benthic oyster population and you want to look at the effect of that as it grows and dies and that kind of thing, most of the early models, the population dynamics models for these kinds of organisms in the past, were population-based models of the average individual. All right, and it's this kind of flow diagram up here where you filter the animal the average animal population filters water that goes to ingestion, assimilation here. You debit that with respiration, and you end up with net production here. Right? That's a fairly standard approach in all of these models. All right, but in this type of approach with the general population model, that's all parameterized for whatever the average individual would be, which we've heard several people say the average doesn't exist. Okay. All right, this type of model, you can put salinity, temperature, and the food field in there, and so you input those as variables, the animal responds, or the population responds to it, and you get solutions or simulations from that. Okay. Now, that kind of model is typically a population model. You start out with, say, the early stage, the newly set juveniles, and you grow it up to the adult. All right, those are it's basically size class-based models. All right, so as the animal grows, it produces somatic tissue, and then also as it gets older, it forms reproductive tissue. And then the reproduction occurs in response to biological or environmental cues of some type. The animal filtration rate sets up this assimilation and respiration, and in fact, that's important. If you key in on what needs to really be parameterized, that's where you look. All right, now, when you start dealing with things much above the level of zooplankton, you find out that the animals gain and lose mass. Right, so they starve, they actually shrink. Right, and that needs to be built into these kinds of models. Is that this net production here gets divided between somatic growth and reproductive tissue, but if the food field or the temperature conditions are bad for the animal, it can't grow and it will actually shrink. All right, and these models are usually in terms, as I said, of energy or carbon units. And because they are size-based models, one of the things you have to end up doing is developing some type of length to weight relationship. The biological literature is full of these kinds of relationships. This is the same species oyster in different parts of an estuary. You don't get the same length weight relationship. Usually you take these, draw a line through the middle of it, and that's your length weight relationship for these models. All right, this is one of the, this is a scale over here where this is now just turned into discrete size classes, and in terms of the biomass and the length of the animal. All right, so it's a nonlinear size scale that you use to go between these different size classes. We'll come back to the implications of that here in a moment. All right, so if you look at the model, let's exclude the the physical environment, the circulation, and everything. You end up with just sort of ordinary differential equations. All right, so net production ends up being somatic tissue, reproductive tissue. It comes from the difference between assimilation and respiration for a particular size class. So now you've got just a time rate change in the oyster here, given by its growth in the somatic and reproductive tissue. All right, but if you're at size class J, you get individuals moving up from the lower size class, and then this one grows and moves up into the next. So you gain from below and you lose to the bigger one. All right, so that's what that is, and that gives you then your time change in that particular size class. All right, now, size class models, right, when you start out with something like this, you start out with, say, 10 animals per meter squared. All right, at the end of your simulation, you still need to have 10 animals per meter squared if you don't put in new recruits. You just grow those animals. All right, now, if you have 10 units of energy formed in size class J, and you move it up to size class J plus 1, that 10 units isn't necessarily one full animal, okay? All right, so what you have to do is you scale the transfers above it and below it by the weight of the animal. All right, now I mention that because if you go back and look in the literature at the early models that were done like this, they weren't scaled. So in effect, what happens is you create destroyed animals by moving them up and down. And it sounds silly, but you really have to, not silly, but you have to conserve the number of animals. Because as far as we're shown, we don't have a lot of governing principles in ecological, biological model, but conservation of mass is one of those. All right, so you have to be careful with this to make sure that even if you're running your model in energy units or carbon units, you still have to conserve the number of animals that you have.
And again, so the spawning and recruitment?
No spawning, no recruitment, okay? This, if you just have 10 animals, you start out with 10 babies and you grow them up, okay? You're not putting in spawning or recruits. All right, you don't have any inputs at all, you just have the same ones. All right, and that's an issue in a lot of the models, older models in the literature, that they weren't looked at in terms of conserving numbers of animals. Okay. All right, so if you then go and worry about some of these other things that I just mentioned, which is shrinkage due to, say, poor environmental conditions. All right, the blue box here is what I just showed you. All right, then you put in resorption. All right, so now you get at something like the reproductive tissue to cover the somatic tissue needs during times of bad conditions. All right, reproductive tissue in a lot of these benthic invertebrates is 30 to 50% of the body weight. So when it spawns, the animal throws away 50% of its weight. All right, humans, when we have babies, we get rid of about 10%, you know. So you can imagine throwing away 50% of your body weight every time you spawn. Right?
I'm sure some would like that.
Yeah, there are people who might have, I don't know, I had a baby, I wouldn't want to do that. All right, so anyway, reproductive tissue counts for that. So once you put reproduction in here, all right, then what you end up dealing with is all of this here. All right, you have to keep track of the gametes and where they go, how viable they are, and that kind of thing. And then you produce larvae. And when you produce larvae, you come down here, and then you worry about larval mortality, recruitment events, and all that. And then the larvae come back and provide new recruits. All right, so that's how you get new numbers of animals. So once you start doing this, you end up with a lot more complexity in what goes on here, and a lot more things to parameterize and to keep track of. And then you add additional terms to the equations here, where now if you can shrink, then the big guys come back, and the little guys gain from that. So you're now moving animals up and down in this size class gradient that we were talking about here. All right, so the kinds of simulations you get from this kind of model are shown here. This is a 4-year simulation. Size classes over here. What's plotted is the log of the number of individuals here over time. And the gray stuff here is the development of the reproductive tissue. All right, so if you look here, there's a buildup of reproductive tissue here. This decrease in the population, remember this is in terms of energy here, or the size. This decrease is the spawn. So you can actually see the loss of the energy, the loss of the mass as the animal spawns. So you can track the spawning events through this whole thing. You also get the range of size classes that are reproductively active, the ones that are producing recruits that go back into the population. So this gives you the time history of a lot of things, gives you the change in the population structure, and that's what you get from this kind of model, and that may be adequate for a number of applications. But the limitations here are that you're looking at the average individual. So it doesn't allow for variability in things like reproductive rates, respiration rates, those kinds of things. The larvae in this kind of model are a loss of mass for the post-settlement population, and then they're basically a gain of mass to the smallest size class. So what they're doing, you're just sort of adding and subtracting mass. You're not changing the characteristics of the population the way you might if you put in larvae that have a range of genetic variability. All right, and the way the model is developed here is if you gain or lose weight through a spawn, it requires that you gain or lose length because it's based upon a weight-length relationship. All right, and that's a big limitation to these kinds of models because it's not realistic. You can lose weight, but not lose length, okay, as an example.
So where does something like oxygen come in? Do you have any limitations?
Oxygen limitations are built into these models here. Right, because these animals live in environments that tend to have hypoxia or anoxia at times of year. So that's a huge one. Okay, So let's go look now at these kinds of models with the recent past. That's what it's called here. All right, so now we go to the individual. We put that in here, use an individual-based model, and then take the individual and through some magic develop a cohort by varying characteristics along a range of variability, and then somehow magically take this cohort and build up a population. All right, so what we want to do here is use an individual-based model to look at ranges of variability, build up a cohort where you have individuals that have phenotypic variation, and that variation is determines the population response to the environment. And in this type of model I'm going to show you here is that if age, which is a measure of how old the animals are, is that the age and where length is sometimes used as a proxy for age in these animals, that is decoupled from the weight, right? So age and length are now actually decoupled, right? All right, and so why is that? If you look at how animals grow, and this could be anything, it could be humans, it could be any animal. This is the length axis here, this is the age over here, so this is the age-length distribution. And if you look, the reason that the other one I was showing you has a problem is that if you look at a given age here, you see that that age has associated a range of lengths. All right, you look around the room, you find people who are your age, you're not all the same size. So there is variability in length with age. And if you look at length can, go over a span of ages. The same length goes over a span of ages. All right, and that range represents the genetic variability within the population. And you get these nonlinear curves to these age-length distributions because most animals hit a maximum length, okay? And, we all grow that way. We seem to be able to continue to increase in mass, but, length is, or height in terms of humans is finite. So this type of relationship tells us that, what we were doing in the other models where weight and length are directly coupled isn't a very good way to do things. All right, so now if you look at a model that was developed for a hard clam, what's shown here, again, you go through the thing, you have the environmental forcing here, filtration, assimilation, respiration, get down here to net production, apportion it between somatic and reproductive tissue, and then if the somatic tissue is positive, you can in fact gain weight. If it's negative, you in fact lose weight. But what we do here at this point is use a concept which is scope for growth, which is if you've got enough left over after you've done all your maintenance, you've done everything, then you can increase in length. Right? So this is a condition index thing. So you look at the animal's condition. If it's positive, then it can gain in length. If it's negative, it can't. Or if it's neutral, it just stays at that length. So that's a different approach here. So there's a different calculation that allows the length and the weight to be decoupled. Reproduction still goes through spawns, eggs, recruits, and then back into the cohort. All right, so by doing this, you can actually allow the animal to gain weight but not reproduce.
Is your mortality constant?
In this example I'm showing you, there is no mortality. It doesn't have to be. You can do whatever you want it to do. Right? But there is a mortality here to the recruits. There's a planktonic mortality. There's a mortality that's due to the fact that there's genetic abnormalities in the gametes, which is why you track the gametes. I'll come back to that in a moment. Okay? All right, so in this kind of a model, you still calculate, but you're calculating differently. You look at the time change in the weight, you then go through the condition index, and then look at the time change in the length. So it's a little bit different approach to how you do this kind of model. Okay, so what do the solutions look like? Well, this is an example of a simulation in weight and length space that was done for a hard clam, mercenary, if people are interested. Right, the dotted line here shows the absolute maximum length and weight that one of these animals can get. The dashed line here is sort of what's the standard, and this line is the simulation. So it's not a nice smooth curve now as it was in the previous ones. So what it does is you see it going up and down, changing, going up and down, and that type of thing. So you can get an increase or decrease in weight without a corresponding change in length. And if you look at these kinds of things, these are the spawning events. Right, so this animal spawned right in here, that's a big spawning event. There was no corresponding change in length. Right, and that's a big advance, I think, in how these kinds of models can be done. And it's not just these kinds of animals, it's pretty much any organism that you're growing up in these size class models.
What's the trigger for spawning? Seasonal?
The spawning cue for shellfish, there are two parts to it. One, there's a temperature, apparently a temperature cue, so that when it warms up. The other is when the animal reaches the point in an oyster when the reproductive tissue is at about 40% of the total body mass, right? And you need those two things to happen, and when they happen, the animal spawns. Okay, right? So if you look at then how do you do something with this kind of model, or how would you use it in the context we're talking about here? All right, well, we can look at the effects of environmental conditions on clam weight and length. All right, and this is a simulation up here of the dashed line here is clam length, this is weight, and this is a sensitivity simulation that was run with a period of starvation imposed in years 3 to 5. Right, starvation would be something like environmental conditions were such that the phytoplankton bloom was delayed, there was lower phytoplankton production in the year, something like that went on in here. So you impose that through the environmental conditions, and then you look at the effect of that on the growth. Right, this is the relative condition of the clam, right, and for the two cases, it's a dashed line and the solid line here, where it's a standard case and then the one with starvation. One thing is that the condition of the animals change over time, and what happens is the animal gets older, it spawns less frequently. There's some biological or ecological implications of that. But the management part of this is that if you look here, all right, this is an animal that's 1 gram, all right, and in this simulation, it reaches that weight and length here in about two years. Turns out this is about the size that they start harvesting these animals. Right, during the starvation period, an animal didn't get to 1 gram until about a year and a half later. Right, so this model has been used to look at things like what happens under changing temperature conditions, changing food quality, changing food availability, food quantity, and the application of this model was to hard clam populations in Great South Bay, where apparently the phytoplankton community has shifted from large cells to small cells, and the clams grow poorly on the small cells. So it's looking at things like this. So when do you open the clamming season? When do you close it? How do you set the quotas? All right, now, as I said, this is an individual-based model, so how do you take this and go to something that's more cohort and population-oriented? Well, in this case, it's a very ad hoc way of doing it. You take something like a Gaussian function to look at, and you take a couple of characteristics of the individual, like respiration rate or assimilation rate, and you vary them over a range around the results you get from the individual-based model. All right, so you do that, you get this large, you run it's all post-processing in effect, looking at if you have this individual and you know these two biological rates vary in this way, can you sort of fill in a matrix that would give you a range of variability around that one individual? All right, so you do this and you pick the characteristics you want, and you build up a series of cohorts based upon the spawning events, which are usually 1 or 2 per year or season. All right, so if you do that, you get sort of this kind of picture here. This is just an example, all right, where the respiration rate, the first simulation would be here at 1, all right, where that would be the one individual-based simulation. And then dairy respiration rate, this would be, double the value at 1. This is half the value at 1. And then this is the initial egg size. And you vary egg size across here from all across these ranges. Right, so if you look at that, then you find out that this is the area here. This range of egg sizes and this range of respiration rates are the larvae that will be successful. And success here is defined in terms of being able to reach metamorphosis and complete metamorphosis. So this is the range that's successful. The rest of these all die or are not successful for a variety of reasons. Right, it turns out that this is actually an example for an oyster, not a clam, but it turns out that oysters' eggs, their initial eggs, are somewhere between 40 and 70 microns. Right, so you can quasi-validate this by looking at things, sort of non-quantitative types of approaches, but it does allow a little bit of checking on this. Okay. Now, this is an example of a cohort structure that was developed from that model, from the spawning events in a year, and you can see the cohorts change with length. This is some characteristics of these cohorts. So the biomass is going down as the animals age and die. All right, and you vary these various rates. You can choose whatever you want to do it, and then construct cohorts with this range of variability. Because if you look here in terms of length, now there are animals in this cohort that are going all the way from, say, 15 millimeters up to about 25, 30, somewhere in here. Right? And then you take that information, you use a broodstock recruitment relationship to construct populations from those cohorts. Right? This is all done as in an ad hoc way, post-processing. And that's how you get these age length distributions, population distributions, and population over time, or population length over time. So it's getting a step closer to reality and to where it can be applied to things. But the limitations to this type of model is that the population variability is based upon setting up this probability distribution. It's Gaussian, and it's in sort of an ad hoc way of doing it. And you can't determine cause and effect. All you can say is if respiration rate is higher, this animal does better under these conditions. All right, so the introduction of new traits or modification to existing traits is not possible in this kind of model. And what we really want to be able to do is just track genetic variability of these populations. All right, so we now look at these kinds of models, and this is what I'm calling the present, where we are now in our understanding. Is you put in the genotype back here. All right, so you do this through knowing how many chromosome pairs there are, the number of chromosomes. You can specify loci, the number of loci per chromosome, or multiple alleles per gene, however you can say it. And so now, what the way things are going with these kinds of models is to integrate the population into genetic processes. All right, so now you have a genetically determined phenotypically based physiology, and the phenotypic response is now modified by the genotypic constraints. And the phenotype is what's responding to the environment. So now we have an additional piece over here, which is the animal genotype, or the genetic structure of the population. All right, so the example I'm going to give here is based upon some oyster models. All right, and this goes to what I said before, is we have to understand the biology of these organisms to really do this kind of model. All right, oysters have high fecundity. Each spawning event is 10 to the 6 eggs or more. And in some areas where there's warm areas, like along the Gulf of Mexico in the United States, where it's always sort of warm, there can be multiple spawns per season. At higher latitudes, there may be only 1 or 2 spawns within a season. All right, oysters are very annoying animals in that they're protandric. All right, so they're males when they're young and at small size, and then as they get older and bigger, they become female. All right, and so you have to track, and if you're going to do a genetic model for these animals, you have to track the sex chromosome to know male, female, male, male, or female, male. And it's the female, male chromosome, or it's paired, that gives the protandry. All right, so an implication of that is the sex ratio of the population changes as it ages, right? And that's a problem because the fishery targets the large animals, which are all female. So there's some areas in the U.S. estuaries where the populations are becoming all male, right? And that's not good because you still need the female to do the reproduction, all right? And so it's an issue in terms of management. The other thing is that oysters have a high load of lethal mutations, right? And that's classic of animals that have high fecundity, all right? Lots of animals do that. Why they do it, I don't think anybody knows, but it doesn't seem to matter because some of them don't have these lethal mutations. All right, and then there's this issue that's come up now in terms of marine diseases. It's called Sweepstakes reproductive success. And what that means is that they're in a large population, like a whole bunch of oysters or clams in a bay or something, there are only a few members of that population that are actually contributing the progeny that's maintaining the population. All right, so if you have an area that's actively fished, you don't want to take out all your big producers or whatever, but you don't know who they are or where they are, and who it is changes with time and everything. So this is something that's being looked at a lot now because in a lot of these areas where these diseases have come in and wiped out the populations, the bays are repopulated, and when they're repopulated, the genetic structure has been altered because of these sweepstakes winners. They're the ones that survive. It's the equivalent in Europe in the 1000s when the Black Plague came through, and the people who survived that ended up, looking genetically, or their immune systems were different, and then their offspring ended up with stronger immune systems, if you believe what you read in the literature. Okay, so there are a bunch of things to look at here. All right, so if you put in the explicit genetics, what you can actually do is track individual alleles and end up looking at things like genetic drift and the loss of those. You calculate the effective population number, which is the number of individuals in a population of, say, 1 billion oysters. What percentage of that is actually contributing the progeny to the population? And you can look at the introduction of new genetic traits. All right, now that's important because if you've been reading about the increasing pH in estuarine systems, there are a number of papers come out recently that show that larvae of clams and oysters and other estuarine species don't do well at lower pHs. All right, there's also, and that's something of interest now, is whether there can be some type of adaptation. Disease resistance is another one, and then just general warming temperature is an issue. All right, so now that I've said all this, how important genetics is, can we actually do it? All right, well, what this shows is down here. This is from Shmeen Woe's work, who's an oyster geneticist. This is a map of the genetic structure of an eastern oyster. The red things right here are locations on these genes that he's identified as conferring resistance to a particular disease. All right, so this particular set of oysters, or the oysters from this particular area, apparently have the genetic structure that they can survive a particular disease that affects oysters along the east coast of the United States. So what you would want to know is, how would we be able to transfer this particular genetic trait into these populations? Can we modify them? All right, so these are some simulations that just to show you. I'm not going to go into detail here, but these are 100-year simulations. These are 2 alleles on the genes. Just to show differences here, this one remains pretty stable in the population, changes a little bit. This one goes up and down. These 2 become extinct after 50 years of simulation. So you can follow these individual traits, and then they go into the progeny and look at establishment of traits in populations. All right, as an example, what's shown here, this is an application that was put in. If you add
had the number of immigrants here, and you want to establish a particular trait in your population, what this simulation is indicating is that if you change over 25% of the population each year continuously, and you're putting in this many immigrants here, this tells you how many years it's going to take to establish a trait in the population. If you do it first in the larvae, just by putting in larvae or by putting in the adults. Right, the reason the adult is higher here is because the adults spawn more than once. All right, so this is a continuous, these two are continuous. This is if you do episodic inputs. All right, the reason this is of interest is because there are large programs. All right, I showed you that there are oysters that have disease resistance. All right, one of the management strategies for disease is to grow those oysters in a hatchery and put them into the bay, and hope they become established and they eventually become the population in the bay. What this says, that's a sustained effort over tens of years to do that, to introduce a new trait into a population like this. Right. This again is just an example here of allele frequencies on individual chromosomes, and again tracking them over time. You can see some of them go extinct, some of them have large fluctuations in the state in the population. All right, so if you do look at those kinds of things, all right, how do you then do this? All right, well, this is again an oyster, the shellfish on the bottom here, the post-settlement population, the larval phase here. The adults spawn, the larvae carry the genetic trait, they move around in the population, and then they set somewhere either on their same reef or somewhere else, and these are the ones that are moving around the genetic characteristics. So the larvae just look at as the genes floating around. Right? So what does it take to do this? All right, when you get to this level, start looking at, you invoke a circulation model, all right, where the larvae become particles in this. All right, there's a larval model over here where the larvae grow and they have a behavior. And then there's this genetics model that sits behind everything for the larvae and also for the post-settlement population. So the larvae spawn, they move around, and then they set in the post-settlement population, and that's how the traits get introduced into the new population. So it's a fairly large structure of model that's required when you get to this level. And what's shown on these, I think I probably ran out of batteries or I'm sorry.
Can people yet?
Yes.
I think the battery is gone on this. All right, so this is an example where this was applied to Delaware Bay. The colored areas here are actual natural reefs in the bay. All right, so if you release the particles from these reefs, all right, and this was looking at the introduction of disease resistance into this bay. All right, so if you look at the particles released from these reefs, and you want to you mark the genes with the disease-resistant gene, and you want to know where it goes. What's shown here is a connectivity matrix where this is where animals were released. This is where they set. And one of the things that came out of this is if you look here and here, there's a lot of self-settlement. So animals that spawn on a reef tend to stay on the reef, but there is exchange between the reefs. But down, in the upper parts of the bay and the lower parts of the bay, they are receiving inputs from other areas, but at very small rates. All right, so this says that it's going to take a long time. If you go out here every year and dump oysters with a particular genetic trait onto that reef, it's going to take a very long time for the bay population as a whole to acquire those characteristics. All right, and this is an example of looking at exchange rates in a more general way from the upper bay to the lower part of this particular bay along the salinity gradient. And if you look at this kind of idea where you use the circulation larval model to calculate the connectivities between these areas, and then start looking at how you introduce the characteristics. Right? Because I just got through saying larvae are the ones carrying the traits. All right, so now if you do that, you start saying, well, how long is it going to take to introduce the traits from the larvae? All right, well, this tells you the frequency of the introduction over time. And if you look at this, you say, oh, the larvae are not very good at moving things around. All right, so they're not really introducing these alleles very quickly. But if you look now at the adult population, the post-settlement population, you find out that, yeah, they're actually pretty good at getting these traits introduced into the population. All right, the reason is that the adult mortality is what's controlling this, because the adult mortality determines how many adults there are to make the larvae. All right, so this is an important part of what goes on in these phases. It's not just the larvae, it's what happens with the adult population, how long they're there, and how long they can continue spawning and producing larvae that eventually do transfer it. Because the larvae do transfer the trait, but it's the characteristics of the larvae as well as the adult population that matter. Okay, so last couple of things here. I said at the beginning that the estuaries have a problem with carbonate balance. Okay, shellfish have a shell, right? So in addition to, this is a simplified version of all that I just showed you. This is the growth and the death of the oyster larvae, the oyster post-settlement population. All right, so this is a simplified form of all the modeling structure I just showed you that goes behind that. This is a shell budget up here. And the way you get shell is that the animal dies, the shell stays on the reef. There is a regular dissolution of carbonate shell by seawater, just burial and that kind of thing. But the addition comes from the dead individuals. All right, so this is a shell budget model in a very simplified form here. All right, so the shells provide a habitat because they give the larvae a place to set. All right, and they're very important for the habitat of the general estuary and coastal waters. All right, so what's shown here is a simulation that says this is the shell mass that you get with this particular mortality rate here. All right, so high mortality here, gives you, it's counterintuitive because the high mortality is giving you less shell. All right, but what happens is that the shell is formed usually by the larger animals when they die. All right, and so if you have a lot of large oysters here with a low mortality rate, you're actually putting in more shell because out here at the higher mortality rates, there are not very many large individuals. All right, now this tells you something about what happens to the large oysters and the shell mass that goes into the estuary as a function of the mortality rate of the large oysters. All right, so out here you're not getting a lot of shell even though there's a high mortality rate. So you want to keep the mortality rates back in here so that the large animals are dying at a rate that it maintains themselves, but is also putting a lot of shell into the estuary. But the problem is, this is the fishing mortality. All right, so by fishing the shellfish out here, what it's doing is it's having an effect on the shell that goes into the estuary. All right, and this model is now actually being used to look at shell repletion rates because a lot of states in the U.S. say, well, we'll just buy up shell and dump it in the estuary. All right, well, it's extremely costly to do that, and in fact, it might be more cost-effective to take this red arrow and shift it back up here. Okay, so in conclusion here, I realize I'm between you and lunch at this point, we have the understanding and the tools to allow us to look at the interactions of ecology, biology, and genetics now. And as I said, this is done in terms of a benthic shellfish or a benthic invertebrate, but the application of the approach applies to pretty much anything. All right, so these models are extendable to other invertebrate species, and I would also maintain all invertebrates. All right, if you have the understanding of the species and you have the data sets, then this provides a framework for looking at this, and it looks at the combined effects of environment, growth, and behavior, and the genetic part allows looking at the projection of effects of climate change, because the genetics is the important part of the adaptation to that.
types of complex models to develop the parameterizations that go into the larger models. And I think Marsh made the same comment, is that you want to do the detailed, complex kinds of things so that you know how and what needs to be parameterized to go into the next scale model. And these models are sufficiently robust at this point to give useful inputs for management. As I said, they're used for things like shell repletion experiments, looking at how many animals you need to put in what time period in order to introduce genetic traits into the populations. And the genetic basis allows consideration for the adaptation part. So I'll stop at that point and ask if there are any questions, and I thank you for your attention. Thank you.
Does the model feed on real data like oxygen and temperature and so on? And so that is causing the wiggles in the seasonal cycle.
Okay. The model, that was the, okay, with this, all right, the circulation model there, it gives the environmental temperature and salinity. It also has, which I don't have on here, it has a lower trophic level model in it. All right, so it gives a dynamically evolving food field. All right, it could also have oxygen in it. We don't have that, that's not in there now, because the applications we're using are in areas where oxygen is not, low oxygen is not an issue. What we're trying to do right now is put pH to look at the effects of changing pH on carbonate balance. All right, so all of that is in there. All right, so we don't have time-varying observations of those fields, but we have enough observations that we can evaluate the lower trophic level model that goes into that.
A couple of questions. So there's some assumption made in the feeding. I mean, when you said the size class of the phytoplankton affects the growth rate, they are filter feeders, so you are somehow going directly from phytoplankton and from reproduction to growth. Is that efficiency factor?
Yeah, there's a growth efficiency and all that. Yeah, the interesting thing is that everybody, if you look in the literature, the literature indicates that things like Benthic invertebrates, the shellfish and things, are never food limited because there's always plenty of food. But it's not the right quality of the food, which I haven't even gone into that. Right, and that's where the shift between the large and the small phytoplankton become important, because the food quality is not what the shellfish eat in that particular case.
And when you set these larvae at different heights, do you get like geographic speciation?
Okay, yeah, and in fact, let's see. In this example, this is Delaware Bay, which is on the east coast of the United States. If you look at number 1 up there, it's genetically distinct from 2 and 3. The 2 and 3 are genetically distinct from 4. All right, and that's based upon, again, Shmi Wood's genetics work, where he's gone out there and looked at those animals. All right, and the distinction is a gradient in resistance to disease. All right, where the ones in the upper bay have no disease resistance at all. Mid-bay is marginal, and the lower bay is where the animals have disease resistance. All right, so the management of these species is to try to take number 4 there, move those animals plus hatchery-grown animals to 2 and 3 to develop resistance there. All right. So, yeah, you can see within a small bay like Delaware Bay, there is genetic distinction among the population.
Your disease pressure depends on salinity?
Disease pressure depends on salinity, and the upper bay is what's called
called a disease refuge because it's low salinity and this particular disease does poorly. So one of the other applications here is that in theory, you would never develop disease resistance in the bay because number one, if you look at the arrows, exports larvae, right? And as it's exporting larvae that don't have disease resistance, they're going in and diluting the animals that have disease resistance. Right? So, all right, if you were to go at an extreme, you would tell a manager, don't bother putting in disease-resistant oysters because you will never develop disease resistance. But that's not the answer they want. It's one of those, what you're talking about with the human dental image. You know, all we have to do is put them in and they work. Yes.
Presumably, being reef builders, they're rather keen to be in sort of clusters where there's likely to be like a neighbourhood dependency on the characteristics that are there. For instance, the adults that may be growing strongly might create micro environments that allow individuals to prosper rather than being so stuck, isolated from the rest of the cluster.
Right.
Is that possible to bring that sort of dependency into an model?
I'm sure it's possible, and this model doesn't have the resolution to allow that, because we have a reef.
So the reefs are considered to be literally homogeneous within that sort of
Within
environmental
yeah.
spectrum?
But there are people who are working on habitat types of models where they actually look at the physical structure of the reef, and they try to do that. They try to look at the variability in the reef and what that does for the reef building animals, but also the animals or the organisms that depend on the reef for habitat. Right, but this model doesn't have a resolution to do that. Yes.
Do you know what is the reason for this? What is the ecological reason for this reduced connectivity? What is the reason for this bispeciation? Why is there different haplotypes, different types of shellfish? The meaning of different adaptations.
You mean
Why is it caused? Yeah, what was it caused?
why do the differences in the genetic population occur? Is that, yeah. It's due largely to the salinity gradient in the estuary, and I'm talking here in terms of disease resistance. The salinity gradient, the oyster and the particular pathogen respond differently to low and high salinity. All right, and it's the trade-off between that ends up setting up these different genetically structured populations.
So can it be caused artificially on like pools? Can it be caused artificially and then you can remove them to those specific
Okay,
sites?
you don't have to do it artificially. Delaware Bay did a nice natural experiment for us in the 1980s where there was severe drought. And the severe drought allowed high salinity to persist all the way up into the upper reaches of the bay. All right, and what that did is the disease took over the entire bay and pretty much killed something like 80% of all the oysters in the bay. And the people who worked on that were able to look at how the bay repopulated itself. And it turned out that that was the equivalent of the Black Plague in Europe because the animals that repopulated the bay had a different genetic structure in terms of disease resistance. But it's easier to let Mother Nature do the experiments and no impact statements.
If someone were to try to extend a model like this to a global model, by the form of a parameterization, what do you think are the most important variables that would be sort of
Well, I would turn that around and say, what is the question the global model is asking? All right, is it carbon export from the bay? As an example, if that's the case, then all you need is a parameter. All you need to take from this is probably the parameterization for the filtration rate. You know, so it really depends on what it is. All right, if you're asking, can the disease in this bay spread along the east coast of the United States as global warming it expands the potential habitat? All right, that's a different question than what you would want from that is the export from the bay. either the larvae or the pathogen. Is some understanding of that? So it really depends on what you're asking.
So this type of genetic modeling is really telling you like how the type of traits you want to put in are dispersed. But how about a natural, like how long it takes for the natural system to actually have a genetic modification?
Right. I can give you a paper where we've actually looked at that. Right. Depends on what the.
What was the question, sorry?
The question is, how long would it take for the natural system to establish a new trait?
Actually show up some mutation that
Yeah.
to develop naturally some resistance.
Yeah, like if you develop a mutation that allows adaptation to lower pH, as an example. You can set up those kinds of simulations and look at, if you put something if you set up the genetic structure in such a way, the genes are there and you have a marker for that, and it's a rare gene in the population, you could look at how long it takes for that to get established or to become part of the population, part of the general genetic structure. I can show you a paper that we've done where we looked at some of those kinds of.
Would that be sort of species specific?
In this case, yes, it is.